Hi, everyone. My name is Daniil Gataulin. I'm one of the senior research analysts here at Chardan covering biotech, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Stephane Boissel, the CEO, and Daniil Chung, CMO from SparingVision, to the second series of our virtual ophthalmology summit, where the focus is on retinitis pigmentosa. Welcome, Daniil. Welcome, Stephane.
Hello, nice to see you again, and thank you for having us today.
Yes, our pleasure. Yeah, so to start things off, can you spend a few minutes and provide a brief overview of SparingVision and its programs?
Yeah, sure. So SparingVision is an ocular genomic medicine company. And more precisely, we develop a suite of assets that are in vivo product based on AAV, and which are either gene therapy or CRISPR-based product. Our lead product, called SPVN-06, is in phase 1/2 clinical development. We've recently announced that we've dosed successfully and safely the first cohort. We are in the second cohort today, out of a 3-cohort part 1 of the phase 1/2, and we should be in the part 2, which will be a control study sometimes in 2024. Our second product, SPVN-20, and we will speak to those 2 programs in a short while, is expected to be in clinical development next year. And for the rest of the portfolio, it's more early stage, and that include a partnership, a strategic partnership with Intellia Therapeutics that was formed in 2021. And based on that partnership, we have access to Intellia CRISPR toolbox to develop up to 3 products based on their genome editing platform. So that's us in a nutshell. Again, only ocular, only genomic medicine, and only in vivo product based today on AAV only.
Got it. Thank you, Stephane, for that overview. Before we get into more into your program, can you give us an overview of retinitis pigmentosa as an indication? Maybe cover what stages there are and what stages your company targets, market size, and current therapeutic options.
Sure. Maybe Dan, you are the best person to start answering this question, and I may add a few things up.
Great. Thank you, Stephane. Good to be with you, and thank you again for your invitation here. So retinitis pigmentosa is the most common form of inherited retinal disease. There are about 70 to 80 different genes that make up this group of diseases. It occurs in about 1 in 4,000. So in the United States, conservatively, we're talking about potentially 80,000 patients that are affected with a form of RP. So RP is a disease that is progressive in nature, and it really starts by the generation of these outer light-sensing cells known as rod photoreceptors. They're for peripheral vision, nighttime vision, things of that nature, but then it progresses to central vision. So basically what we use in our daytime for color vision, our best clear acuity, and eventually those central cells that give us that visual function, those also start to die. So the idea is that with these programs, we're hoping that we can prolong the life of those central cone cells to give us more activities of daily living and in keeping quality of life for these patients with rod RP, which is a form of rod cone dystrophy. And there are other forms of rod cone dystrophy other than RP, so those are also things that we can look at. Now, when it comes to the whole idea of treatment options, there is really only one treatment that is approved for inherited retinal disease, and that is regimen parvovec, otherwise known as Luxturna. But even this is really more for an early form of inherited retinal disease. A lot of times it's commonly referred to as Leber's congenital amaurosis. There aren't many cases of true adult onset RP that have RP65 as the gene result. Now, being gene agnostic, we really can broaden that funnel of who actually could benefit from this, and it's not just gene specific. So as RP65 is less than really 1% of the adult RP population. So there's still high unmet need. And we're really based right in the middle of when these rod cells really start degenerating. It's a little different than the single gene replacements. And then we can also intervene with another product when these patients are at end stage, but not quite as end stage for when you need to use stem cells or optogenetics or something like that. So we're right in the middle of these other two relatively common modalities.
Mm, got it. Yeah, and that's actually what, what was one of my questions. There are, as you mentioned, several approaches in development, including optogenetics. Obviously, we understand the difference between optogenetics and your approach, but in terms of others that are in development, what do you think makes your program unique? And maybe to take it one step further, what gives you confidence in the success of your program?
So there are a number of things that you can look at. Number one is the science of RDCVF has really been explored by a number of different groups, and there are quite a few publications that have shown the efficacy in preclinical models of this, whether it's using the different forms or both forms of the RDCVF. And that's been done by many different labs, not just those associated with our founders. I think the one thing that gives us promise in this is that it really exploits a pathway that already exists in all of our eyes. RDCVF is in a mechanism that is secreted by the rod photoreceptor cells, and it binds to the cone photoreceptor cells, and really this pathway to create the longevity of the structure and function of the cone itself. So we're really just exploiting an innate pathway that already occurs in the eye in the sense of a gene agnostic approach, because for us, we're not trying to treat the underlying genetic cause. We're simply saying that we're replacing the product that is missing because the rod cell is gone. It doesn't matter why the rod cell is gone, simply that it's not there to produce the factor. So through gene therapy, we're simply replacing that and getting that protective effect again. So,
and if I may add something to what Dan said, so RDCVF, as you know, is one of the two transgenes in SPVN06. And you mentioned optogenetics, but this is not exactly where we compare our product or what we compare our product with. SPVN20, the second asset, is much closer to optogenetics, since SPVN20 will aim at restoring function in late stage patients who have lost most of their visual acuity. SPVN06 on the RDCVF biology will come much earlier in the evolution of the disease, and possibly through a significant spectrum. As long as you can find cones in the retina, the idea would be to protect what is left of the vision at the time of intervention. So it's totally different concept than optogenetics. Again, I wanted to make sure that you understand SPVN20 is much closer to optogenetics, which at the end of the day will come at a much later stage in the development of the disease to possibly rescue some function.
Got it. That makes sense. So there are always some benefits of not being the first one in development, and having some programs ahead of you. Again, not necessarily closely competing with the same approach, but just in general in the indication. What are some of the learnings that your company took from those other programs?
Dan, you want to give it a start?
Sure. So when it comes to learnings, obviously the things that we can learn from some of the safety data that has been developed in prior gene therapy experiments or gene therapy trials just on the subretinal approach, and how that has a favorable safety profile, both in the immunogenicity involved as well as the technique. So we've looked at that. Obviously, we've adopted some of the parameters, such as pre-dosing with oral steroids to minimize inflammation. So those are some of the things that we've learned is really on the technical side of things. When it comes to the actual idea of a gene agnostic approach, there really hasn't been a whole lot done in the sense that for going into clinical therapy. There are others that have been tried. But again, one of the unique things about us is we're really trying to exploit an innate pathway that really already exists to maintain cone function. So those are things that we really kind of took to heart.
Got it. Understand. And so to spend a little bit of time to talk about SPVN06 construct itself. So I understand it carries both RDCVF and RDCVF-L transgenes. What is the significance of having both of them?
Yeah, each one has a slightly different effect of maintaining the function and structure integrity of the cone photoreceptor. And as we said, the cones is what we use for daytime vision, your best acuity, and the last thing to go in RP patients. But the RDCVF, the whole construct, is really made from the nucleorhodopsin-like 1 gene, the NXNL1 gene. And it actually has two different parts. There's a short form. This is actually secreted by the rod photoreceptors, as I had mentioned before, and it binds to the cone photoreceptor by a membrane receptor called basogen-1. And this in turn complexes with a glucose transporter, and this increases the amount of glucose that actually comes in to that area from the choriocapillaris. And this in turn promotes aerobic glycolysis. Which then in turn increases the carbohydrate formation. And this is really what stabilizes the cone function and structure. Now, when you take the whole gene, which is only 2 exons or 2 coding regions, this is more of a powerful antioxidant that occurs. And this is not secreted, but is found both in rods and cones. And so, as you may know, the retina uses more oxygen per gram weight than any other organ in the body. So you really need to mitigate all the issues associated with that level of oxygen usage. So when you put these two together, the neuroprotective effect of the short form and the antioxidant effect of the long form, you put those together, you subsequently get the protective effect to the cone function and structure.
Got it. That makes sense. And what vector do you use in your programs, and how did you choose that vector?
So we use an AAV, and basically we use an AAV that has good affinity for photoreceptors, but still has some transduction efficiency for RPE cells as well.
If I may add something to what Dana said, this is an AAV that we do not disclose, but which has already been tested in many different clinical trials, including ocular trials, at a much higher dose. So we feel confident that when it comes to safety, at least the AAV is not going to be an issue.
Got it. Okay, I understand. In terms of preclinical data, can you provide some of the highlights about SPVN06 that gives you confidence in the clinical outcomes?
Sure. So the whole biology of RDCVF and RDCVF-L is not new. It was started by José Sell and his group in Paris more than 20 years ago. And so over those 20 years, they have generated multiple proof of concept on various animal models. So it will be fastidious for me to summarize 20 years of research in 1 minute. What I can tell you is that when it comes to the pharmacology, we've used a few mice models and one small piglet model. And in everything that we've touched, we've seen an impact on either the structure or the structure on the function. So we are very confident when it comes to the pharmacology that RDCVF is doing something. Of course, we'll see what it will be doing in human in 18 months from now. But for pharmacology, we have, again, a strong corpus of evidence based on preclinical models. In terms of safety, we've conducted primate studies across 7 different doses over the last 2 years. And we found a sweet spot where we have a good safety profile, a good safety margin, and a very significant pharmacological activity. And this is with that sweet spot that we've started the human testing. So again, it's very difficult to summarize in 1 or 2 minutes all those years of research, but we went into the IND and CTA process and into the start of the first in-man with a very significant package of compelling data supporting testing in human.
Yeah, got it. No, that's fair. And so you launched your phase 1/2 trial, Prodigy trial, right? Earlier this year. You just reported your safety data from the first cohort of 3 patients. So in terms of the trial overall, can you talk about the design of the trial, the patient population that you're targeting, and when you get into efficacy analysis, what efficacy signals will you be looking for?
Maybe then you should. I will add anything there if I feel appropriate.
Great, thank you. When it comes to the trial, so this is obviously we were looking to have a trial that would be both in the US and in the EU. So we were able to get both CTA and IND clearance, and we also took their comments into strong consideration and basically affected the final design of the trial. The trial is divided up into 2 parts. The first part is what you'll see very traditionally is a 3-dose escalation with 3 patients in each cohort. Because this is obviously a phase 1/2, main emphasis is placed on safety. There are several observation periods within the cohort, and after each cohort is done, we have a DSMB review before we go on to the next cohort, and after they're given us full approval to proceed. So the patient population is we have 3 different genotypes of relatively well-known RPs, and that we are putting those into those first 3 cohorts. Now, part 2, the second part of the phase 1/2, is an extension cohort where we actually have 3 arms. There'll be a naive control arm, and then the DSMB will give us a recommendation as to 2 best optimal doses from the dose escalation. And then they'll be randomized into 2 different intervention arms. So you'll have 3 overall. Then after 1 year of observation, after the last patient is injected, we'll use several different outcome measures, both functional and structural. So functional, we're looking at things such as microperimetry and different types of perimetry, both kinetic and static. We're looking at fundus autofluorescence in terms of function of the RPE cells. We have ERGs, we have full field light sensitivity threshold tests. We are doing patient questionnaires, and then a number of other things on the functional side. And then on the structural side, we're doing OCTs, where we're looking at the ellipsoid zone and looking at this because we did 2 natural history studies where we also looked at a lot of these parameters. We're analyzing some of that data now, and we hope to be able to show some that we think are promising in this next coming ARVO. We're also looking at adaptive optics because another study using USH2A basically showed that there is some movement in the cone density of adaptive optics was able to show. So there are a number of structural things that we're looking at to correlate with the functional as well. But this is a phase 1/2, and obviously there's strong emphasis to make sure that we have a good safety profile. But we're looking at these potential efficacy outcomes for see any trend or maybe a responder effect here and there. So that's kind of what we're going for.
Yeah, got it. And speaking of safety, can you summarize, the safety findings that you just announced?
Yeah, the safety findings were basically self-limited. They resolved without further intervention, and they were relatively minor. There were no serious adverse events. And so the DSMB felt very comfortable after the first cohort to let us go into the second cohort with no modifications or adjustments to the protocol or what we're doing.
Okay, and you mentioned about the steroid regimen in the beginning. Can you summarize what regimen you're using again?
We're using one that's somewhat standard in the industry right now, very similar to what Luxturna used. Basically, we dose 3 days ahead of time. We use 1 milligram per kg by body weight, and then we do a slow taper after the injection. And obviously, at that point, the PI, if there's any modification of that's needed, they're able to do a standard of care but we have not really been able to or needed doing anything like that. So we had very good inflammatory profile in terms of there was very little safety issues there.
Got it. And another question. So there are a number of mutations that could be responsible for retinitis pigmentosa. And being a gene agnostic, that's one of the attractive aspects of that therapy, that you could potentially treat a number of those. But for this trial, are you limiting yourself to specific genotypes?
Yeah, there's
specific variants?
3 genotypes. We are restricting it to just number 1 to get a little more homogeneous population. Plus, we also had the preclinical models to show proof of concept in those as well. But we do hope to see if we can expand that in future trials, because obviously we do believe that this will be a good gene agnostic approach by the mechanism that we highlighted.
Got it. Okay. And in terms of the initial efficacy data from that trial, I believe you guided for 2025, correct? Is that still the goal?
No, that's correct. That will be the second part of 2025. The primary endpoint is 12 months after the last patient the last patient has been injected. And as of today, we expect the last patient to be injected sometime in the second part of next year. So 12 months from next year, from the second part of next year, we'll give some data sometime in the second part of 2025.
Got it. Okay, I understand. So let's shift gears and talk a little bit about SPVN-20 program. So this is your second program that's being developed for stage 3 and 4. And the transgene in SPVN-20 is GERK. Can you talk about this transgene and this approach in general, and why do you think it's an attractive approach for advanced form of retinitis pigmentosa?
Sure, Dan, you want to start?
Sure. So the GERK gene stands for G protein inwardly rectifying potassium channel gene. And basically what the concept is that, as you may know, that the photoreceptors do something called phototransduction, basically converting the light energy that comes into the eye into an electrical response that gets shot through the retina and then to the brain, and that's where vision occurs. So without something called the outer segment of the photoreceptor, that's kind of where all that magic happens. We call these dormant cells because the cells are still physically present, but the outer segment, that functional phototransduction part, is no longer there. And what we have found that in very end-stage patients, that we have these dormant cone cells left in a small percentage of patients. And the idea is that GERK will then interact with the residual cone opsins that remain in the inner segment and something with the cone arrestin. And through this new channel, they can do a hyperpolarization and have a truncated or shortened form of phototransduction within the residual structures of the cone. And so that's the idea behind GERK, is can we restore some form of visual function from these dormant cells that are in more end-stage RP patients or rod cone dystrophy patients, and potentially maybe even early stage cone dystrophy patients. But right now we're targeting end stage RP.
And this is where, if I may add something to what Dana said, this is this product which is more comparable to the optogenetics approaches. The main difference or the main differences being that SPVN-20, so GERK, is going to target the cones where everything is happening to start with, and not ganglion cells or other cells in the retina. Also, and this is one of the reasons we believe we could not only achieve superior impact on visual acuity, but also color vision, that we don't believe any optogenetics approach will be able to provide. The other difference is that we are using a human GERK, and so the transgene is not from it's a different species, and so we expect lower immunogenicity when it comes to the safety profile. And last but not least, given the biology that was described by Dan, we don't expect to be in the need for light stimulation or extra light stimulation. So all that to say that with SPVN-20, we believe that we possibly have one of the most powerful approaches in the world of optogenetics. Not exactly optogenetics, but this is definitely where the playing field will be.
Okay, understand. You recently presented some preclinical data that was at ASGCT earlier this year. Can you provide some of the highlights from that presentation?
Yeah, I can start, and maybe Dan, you can complement. So that was very important for us. It was the first time we had an opportunity to present SPVN20 orally in a big conference, and we had data on the pharmacology side as well as on the safety on biodistribution side. So we had some mice model data, and mice model, and on the healthy monkeys, we've been able to show very nicely the distribution of the transgene around the fovea of monkeys with an intravitreal injection. We've not said it, but it's very important to say that SPVN06 is a subretinal injection, as it was said by Dan, whereas SPVN20 will be an intravitreal injection. And so it was very important for us at ASGCT to show that with an intravitreal injection of a certain AAV serotype, we can specifically and very nicely target the cones that we will want to target, which will be the cone that will be centered in the fovea.
Okay, got it. Makes sense. And with respect to clinical trials, when do you expect to be in the clinic with SPVN20?
Sure. So we are in the what we call the IND enabling studies as we speak. And IND, actually, it will be a CTS submission in Europe, more likely than an IND, is expected to be at the end of the first quarter of next year, early second quarter. So first part of 2024. And then the idea will be to initiate the trial during the summer. In contrast with 06, for which, as we said, there will be kind of some time between the injection on the effect. We expect to be, for SPVN20, able to observe clinical efficacy much more rapidly because of the mechanism of action, because we will be restoring function and not preserving function. So this is why we like to say that SPVN20, despite being 18 months behind in terms of IND readiness compared to SPVN06, should give us clinical data more or less at the same time in the second part of 2025.
Got it. Okay, I understand. And with respect to GERK technology, so you had both ex vivo and in vivo technology, but in July you announced that ex vivo was sold to 10 Point Therapeutics. Was that always your strategy to spin out that program?
Yeah, and actually we did spin it off the day we acquired Gamut. So the GERK program, SPVN20, is the result of the acquisition of a small startup called Gamut Therapeutics that was back in the spring of 2021. And when we bought the company, they had two versions of GERK, so to speak, one in vivo and one ex vivo. And as I said in the intro, the goal of the mission of the company is to develop in vivo ocular genomic medicine. And so from the beginning, we had an understanding with our board, but also with the shareholders at Gamut, that we would spin any ex vivo work into a specific company, which we did. And at one point, 10 Point came to us. They are a cellular therapy company, as we understand it, and they wanted to in-license this technology. So we sold that spin-off to them in exchange of shares in 10 Point and some milestone royalties.
Got it. Makes sense. And with respect to manufacturing your gene therapy products, is that done internally, or do you have external collaborations with CDMOs?
No, we work exclusively today with CDMOs, and I don't expect that to change in the near future. Maybe in the long term, it will change. But no, we work with CDMOs, and actually the company is like a project management company today. We outsource most of what we do. which is giving us a lot of flexibility. And so we are a team of 35 today. And if we were to internalize everything, including manufacturing, we would be a team of, I don't know, 250 or 300 persons. And well, I think that was a wise decision not to internalize everything, given the desert that biotech financing went through over the last 2 years. So we are in a good place. We have a very lean organization and a very flexible organization.
Mm-hmm. Okay, got it. And earlier you mentioned about your collaboration with Intellia. Can you talk more about that and what you said the biggest benefit to SparingVision from that collaboration?
Well, the main benefit is kind of obvious. I mean, Intellia is the world leader in vivo genome editing, and we were very lucky that when we reached out to them in 2021, they accepted the principle of collaborating with us. So we learn a lot every day by the world leader in the space. They not only give us their technology, but they also develop everything that will go into the AAV. So the way the collaboration works is that we SparingVision, decide on which target we want to work. We have a list of targets in the contract with Intellia. Once the target is selected, we sit down with our friends at Intellia and jointly, based on our understanding of the eye and based on their understanding of the CRISPR editing technology, we decide as to what will be the best approach to go after that target. But this is where Intellia is starting to add a lot of value. We design the best possible construct. They take it to their lab, to their factory, and they develop the CRISPR reagent. They validate the CRISPR reagent, and then they send it to us for us to take it from there. So we've selected the first target a little more than a year ago. I hope that we will have some news soon on the second target. And so far, the collaboration is going extremely well, and we are very very satisfied. I'm still honored to have signed this partnership in 2021, and we hope that this collaboration will last for a long period of time.
Mm-hmm. Got it. Understand. And I see that we're coming up on time, but have a couple of more or less general questions. First, can you talk about the company's lead investors? And along the same lines, what milestones do you expect your current capital to carry the company through?
Sure. So we've raised a total of 135 million euro across the Series A and Series B. We currently have a cash runway up until the beginning of 2026. So when we structured the Series B a year ago, it was very important for all of us, including the investors, to make sure that we could finance the clinical proof of concept with 06 and 20, which is the case, as I told you that we expect clinical data in the second part of 2025, and the cash runway is going into the first part of 2026. So when it comes to financing, we are in a very good place. As to the names of the investors, we have a syndicate of 7 investors. Intellia is also a shareholder, but that is a consequence of the transaction we signed with them in 2021. But when it comes to the VC, we have 2 US group, UPMCE, which is the $1 billion investment arm from the University of Pittsburgh Medical Center enterprise. We have Foundation Fighting Blindness, or more precisely, the RD Fund, the Foundation Fighting Blindness. We have BPI France, which is a French sovereign bank. We have a fund in London called 4Bio, a fund in Spain called Izios, and last but not least, Jeto, which was the co-lead with UPMC of the Series B, which is a European fund. And 2 years ago, they've announced the closing of their first fund, which was the biggest first fund ever raised in Europe with $630 million. So we are in a good place with a nice syndicate of investors, with a good mix of pure VC, if I may say so, and let's say strategic with Intelia, UPMC, or FMB.
Got it. Understand. And one last question is, what do you think the most underappreciated part or least understood part of the SparingVision story is?
Well, that's a very vast question, Dan. I think today people don't realize the full potential of 06 and 20, which are the most tangible asset in our portfolio, because not only it's mutation agnostic, but it also, this is valid for the two assets, it's disease independent. And so if we manage to prove the biology of these two assets, not only the potential in RP will be very significant. There is no equivalent of that in the world of retinitis pigmentosa. But importantly, we will be able to go after many other diseases. And for example, for 06, and we've said it publicly, as soon as we have some certainty on the safety profile of the product, which will be hopefully soon, like in 2024, the goal will be to start some development in some form of dry AMD, where we believe the pattern of the disease at one point could benefit from a biology such as the one SPVN06. And again, this is valid for 20 with different disease. So to come back to your question, I think the day we prove the biology, we will have some work to do as to explain the world, what is the true commercial potential of this portfolio.
Got it. Makes sense. All right. I think that brings us to the end of our session. But again, thank you very much for joining. I appreciate you taking the time, and like always, really enjoy speaking with you guys. Thank you.
Bye-bye.